Now, go to the faucet, turn the cold water on fully, put your wrist under it, and hold for 10 seconds. Feel what happens. Your heart rate increases immediately, not gradually, but like someone flipped a switch inside your chest. Your breathing contracts. Your lungs want to inhale quickly and shortly, even though there's no reason for physical exertion. Goosebumps appear on your skin, spreading from your wrist up to the nape of your neck in less than 2 seconds. Your shoulders shrug up, your muscles tense, your whole body contracts toward the center without you ordering it to do so. All these reactions happen in exactly that sequence, at that speed, in anyone, without needing to learn, without practice, without thinking. This is a complete system activating, a system that most people don't know they have, until they read the mechanism behind it. The reactions you just felt don't stop at the skin, don't stop at the heart, don't stop at the muscles. They go deeper, down to every cell, into every gene, and they change the way your body operates. You just felt that. 11 minutes per week, no equipment needed. What happens in those 10 seconds starts not in the heart, not in the lungs, but in the skin. Right at the outermost layer of your wrist, scattered in the epidermis are cold receptors named TRPM8, specialized ion channels designed to detect temperature dropping below a certain threshold and convert that temperature signal into an electrical signal. When cold water touches the skin, TRPM8 opens within milliseconds, and that electrical signal travels back up along the sensory nerves into the spinal cord and straight up to the brain at a speed where the brain receives the information before you have time to think about it. The brain receives the signal and does one thing immediately: activates the sympathetic nervous system. The adrenal glands release noradrenaline. Not a slight increase, not a moderate increase, but a 200 to 300% increase from baseline levels within seconds. That's why your heart rate jumps immediately, not because you're afraid, but because noradrenaline just ordered the heart to pump blood faster, to deliver oxygen to the muscles. That's why breathing contracts, because the sympathetic system needs the lungs operating in emergency mode. That's why muscles tense and shoulders shrug up, because the body is preparing for action. Nothing in this chain of reactions is panic. All of it is ordered activation, according to a program written long before you were born. That program exists because your ancestors had no choice but to be exposed to cold seasonally, every year for hundreds of thousands of years. And the individuals with the fastest cold response systems were the ones who survived and passed on their genes. That system has been optimized over such a long time that it doesn't need learning, doesn't need practice, doesn't need any conscious signal from you. But modern temperature control has done something no evolutionary pressure ever did before. It completely removed the activation signal from daily life. The system is still fully inside you. The signal has disappeared. And a system that is never activated is just like a muscle that is never used. It doesn't disappear, but it doesn't develop, doesn't get trained, doesn't reach the potential it was designed to reach. The key point in the first 10 seconds of cold exposure is breathing. Breathing is the only thing in that entire sympathetic reaction chain that you can consciously regulate. When you breathe slowly and extend the exhale, pressure receptors in the lungs send a signal back to the brain that the situation is under control. The parasympathetic system begins to counterbalance the sympathetic system, and the shock response diminishes within 20 to 30 seconds. This is not a meditation technique. This is neurophysiology. A conscious signal sent into an automatic system that changes its output. That's why the first 20 to 30 seconds are the most important, not because it gets easier psychologically after that, but because after that noradrenaline has peaked and the body begins to self-regulate. In practice, a reasonable starting temperature is 50 Fahrenheit or 10 degrees Celsius for 1 minute and gradually increase over time. Starting at 37 Fahrenheit, like some people experiment with, is starting at a level where the cold shock response is too strong to control breathing effectively. And you'll get out of the water after 5 seconds without getting any benefits. One small but noteworthy point. B vitamin deficiency reduces tolerance to extreme temperatures, a mechanism related to neurotransmission and the nervous system's ability to process stress signals. The noradrenaline that just increased 300% in your blood doesn't just go to the heart and lungs. It goes to a type of tissue that most adults don't know they still have, a type of tissue that newborns have a lot of, and modern adults almost never activate because they never give their body a reason to activate it. Your body contains two completely different types of fat in function. White fat is an energy storage depot. It accumulates calories and holds them there until the body needs them. Brown fat does the complete opposite. It burns energy to generate heat, not to create ATP for cells to use, but only to create pure heat. Inside brown fat cells is a protein named UCP1, short for uncoupling protein 1, and its function is to disrupt the normal ATP synthesis process in the mitochondria, redirecting all the energy from burning glucose and fatty acids into pure heat. This is thermogenesis, the body's internal heater, a mechanism for warming from within, without muscle shivering, without movement, without any external action. Noradrenaline from the cold shock binds to beta-adrenergic receptors on brown fat cells and activates UCP1 within minutes. This is why your body starts warming up from the inside after passing the first 20 to 30 seconds in cold water, not because the water got warmer, but because brown fat is burning. But noradrenaline doesn't stop at activating brown fat. It also activates a gene whose effects last much longer than any sensation you have in the tub. That gene is PGC1 alpha, and if there is one single molecule that explains why cold therapy changes metabolism at a fundamental level, it is this molecule. PGC1 alpha is a master regulator, a gene that coordinates the actins of other genes, and its main task is to initiate the process of mitochondrial biogenesis, that is, creating new mitochondria inside cells. Mitochondria are where cells burn fuel to create energy. More mitochondria doesn't mean you have a little more energy. It means the entire metabolic capacity of the body increases, fat burning ability increases, recovery after exercise increases, cellular energy processing speed increases. But new mitochondria need to be nourished, and this is where a second gene appears. VEGF, vascular endothelial growth factor, stimulates the process of angiogenesis, that is, creating new blood vessels. New blood vessels deliver oxygen and glucose to the mitochondria just created by PGC1 alpha. PGC1 alpha builds the factory. VEGF builds the supply route for that factory. These two genes don't operate independently but work as a pair, and both are activated by the same signal, cold. And there's one more gene in this metabolic chain that few people know about, but whose impact is very specific. PPARG-C1A regulates the expression of proteins involved in the cellular respiratory chain, that is, the mechanism the body uses oxygen in during physical activity. People with mutations in this gene have VO2 max up to 50% lower than normal people, meaning they tire faster, recover slower, and their physical limits are significantly narrowed, right from the molecular level. Cold activates the expression of PPARG-C1A. Doesn't fix the mutation if present, but increasing gene expression helps compensate for that deficiency. Parallel to that entire gene chain, cold also triggers a separate hormone axis. The brain receives the cold signal and stimulates the pituitary gland to increase secretion of TSH, thyroid-stimulating hormone, the hormone that stimulates the thyroid gland. TSH goes to the thyroid and orders the production of more T3, the most active and direct form of thyroid hormone. T3 increases basal metabolism at the cellular level, increases fuel burning speed, increases heat production from within. There are two parallel loops running at the same time. Cold activates noradrenaline. Noradrenaline activates brown fat via UCP1. Brown fat creates immediate heat. And at the same time, cold stimulates stimulates T3. T3 increases whole body basal metabolism. One fast response loop, one slower but deeper response loop, and both are activated by the same 10 seconds of cold exposure. One point to be straightforward about muscle training. Some studies show that cold immersion immediately after strength training can inhibit hypertrophy. The mechanism is post-workout inflammation, which cold therapy reduces, is actually the signal that stimulates muscle growth. And if that signal is suppressed too early, then the muscle loses part of the growth stimulus. But at the level of 11 minutes per week, and not immediately after strength training sessions, this negative effect is insignificant. The benefits from PGC1 alpha, VEGF, PPRC1A, brown fat, and T3 completely outweigh that risk at this dosage. PGC1 alpha, VEGF, and PPRC1A have been measured directly in clinical studies. The optimal cold dosage to activate each gene at maximum levels is still being researched, but the existence of these gene responses is no longer an open question. While PGC1 alpha is building new mitochondria and T3 is increasing basal metabolism, another system is receiving that same cold signal. The HPA axis, that is the hypothalamic pituitary adrenal axis, activates immediately when the body identifies cold as a stressor. The hypothalamus sends a signal to the pituitary gland. The pituitary releases ATH. ATH stimulates the adrenal glands to secrete cortisol. Cortisol rises during immersion. ATH rises during immersion. If you measure these two hormones right while in the cold water tub, you will see them higher than normal levels. And if you only look at those numbers without looking at what happens next, then you would conclude that cold therapy is stressing the body. But what happens after you step out of the tub is the part the initial numbers don't tell you. Cortisol and ACTH not only return to baseline but drop below baseline, and they stay at that lower level for many hours afterward. This mechanism is called hormesis, a biological principle whose meaning is simpler than its name. Controlled stress, right dosage, right duration, does not weaken the system but makes it stronger. The body receives a stressor, reacts strongly for a short time, then readjusts to a level lower than the starting point. Repeat enough times, the body's baseline cortisol level decreases, chronic inflammation decreases accordingly, and the immune system operates in a cleaner internal environment, less disturbed by chronic cortisol. High chronic cortisol is what suppresses immunity, not cortisol that spikes for 10 minutes, then drops below baseline. That is the difference between chronic stress and controlled acute stress, and cold therapy belongs to the second type. When the cortisol environment is cleared, the cellular immune system begins to operate at higher capacity. Killer T cells, that is CD8 plus T lymphocytes, increase after cold immersion. These are the cells that identify and destroy virus-infected cells and cancer cells, not by creating antibodies, but by direct contact and destroying the target. Overall white blood cells increase, meaning the patrolling force in the blood is denser. Blood zinc levels increase, and zinc is not an ordinary micronutrient in the immune context. It is an essential cofactor for hundreds of enzymes involved in immune function, from immune cell proliferation to pathogen destruction mechanisms. The real-world result of these changes can be observed in a specific population group without needing clinical trials to notice. Swimmers. Swimming combines two benefits at once: cardiovascular from exercise and cold water exposure from pool temperature. And the rate of upper respiratory tract infections in swimmers is significantly lower than in the general population. This is not data from the laboratory, but data from real life, from people who are regularly exposed to cold water without anyone calling it therapy. Now to the part where many people will react wrongly after hearing it. Overall antibodies decrease after cold therapy. It sounds negative. It's not. To understand why, you need to understand the difference between humoral immunity and cellular immunity. Humoral immunity works through antibodies, proteins circulating in the blood that attack pathogens from a distance. Cellular immunity works through T cells, attacking directly. In a normal, healthy person, these two systems are balanced. But in people with autoimmune diseases, the humoral immune system is overactive, creating antibodies that mistakenly attack the body's own tissues instead of pathogens. Cold therapy reduces overall antibodies while increasing targeted T cells, meaning less mistaken attacks and better specific defense. It's not the immune system weakening, but the immune system being rebalanced. Parallel to that entire immune chain, cold activates the expression of genes involved in the production of glutathione, the strongest antioxidant that the liver can synthesize. Glutathione not only directly breaks down free radicals and toxins into harmless particles, it also regenerates other antioxidants that have been oxidized during use, including vitamin C, vitamin E, and CoQ10. It is the antioxidant of antioxidants. And if you have a genetic problem with oxidative stress, then cold therapy activating the glutathione pathway is a more important reason to prioritize cold over heat therapy like sauna or hot tubs, which tend to suppress antioxidants instead of activating them. Regarding tumor suppression ability, the mechanism involves brown fat and glucose. Cancer cells have an abnormal metabolic feature called the Warburg effect. They rely heavily on glucose as fuel, burning glucose even when there is enough oxygen, instead of using the more efficient oxidation mechanism of normal cells. When brown fat is activated by cold and absorbs large amounts of glucose to generate heat, circulating glucose levels decrease, creating a less favorable environment for cancer cells. Combined with increased T cells, ready to destroy abnormal cells, reduce chronic inflammation, which is a favorable environment for cancer development, and activation of antioxidative stress genes that prevent accumulated DNA damage, the overall picture is an internal environment less friendly to cancer cells. It needs to be said straight about the level of evidence here. The anti-cancer mechanism in humans is still preliminary, mainly from preclinical data and mechanistic inference, with no large-scale clinical trials directly confirming it. But each link in that mechanism chain is understood very clearly individually. Finally, at the level that most cold therapy users feel most immediately, pain reduction, less muscle soreness, and faster recovery. Cold slows the conduction speed of pain signals along C fibers and A delta fibers, the two types of nerve fibers responsible for transmitting pain sensations from tissues to the brain. Cold-induced vasoconstriction reduces edema at the inflamed area, reduces pressure on nerve endings, reduces pain signals. Muscle soreness decreases because cold slows the local inflammatory process at micro-damaged sites after exercise. Recovery time decreases because vasoconstriction followed by vasodilation when warming up accelerates the clearance of lactate and inflammatory cytokines from muscle tissue. T cells and white blood cells have been measured directly in controlled trials, and the results are consistent. All that has just been described: noradrenaline increasing 300%, brown fat burning glucose, PGC1 alpha creating new mitochondria, T cells increasing, cortisol dropping below baseline, are all reactions that occur on top of a deeper layer that most discussions about cold therapy never touch. That layer is gene expression. And to understand why it is more important than any of the hormone responses just mentioned, you need to understand one fundamental thing about how your genome actually works. Your DNA does not change. The nucleotide sequence in every cell of your body today is identical to that sequence 10 years ago and will be identical 10 years from now. But not all genes in that genome are active at the same time. The environment decides which genes are turned on, which genes are turned off, which genes are transcribed into RNA, which genes lie silent in the DNA strand without producing any proteins. This is epigenetics, not changing the gene sequence, but changing the level of gene expression. And this is why two people with nearly identical genomes can have completely different health outcomes depending on the environment they live in. Winter arrives, temperature drops, the body has to cope. This repeats every year throughout the 200,000 years of Homo sapiens evolution. And the result is a set of genes designed so that cold is an activation signal, not a danger signal, an action signal. A signal that the body needs to operate at higher capacity, repair faster, protect itself more effectively. Then temperature control appeared. Thick clothing, greenhouses, heated cars. And within 70 to 80 years, humans have lived at room temperature of 70 Fahrenheit almost year-round, completely removing the activation signal that the genome has been waiting for. 70 years compared to 200,000 years. The genome has not had enough time to adapt to comfort. Those genes did not disappear. They did not degenerate. They are waiting for the signal that has been removed from the modern living environment. The signal is cold, and these are the genes that are waiting. PGC1 alpha has been mentioned in the metabolic layer as the gene that creates new mitochondria. But at the gene layer, the perspective must be broader. PGC1 alpha does not do just one thing. It is a master regulator, a gene that coordinates the activity of dozens of other genes located downstream in the same network. When PGC1 alpha is turned on, it is not one gene that is activated, but an entire network that is activated, including genes related to energy metabolism, anti-inflammation, protection of cells from oxidative stress, and control of the cell life cycle. One cold signal turns on a master switch. The master switch turns on dozens of smaller switches. PPRG-C1A regulates the expression of proteins that participate directly in the cellular respiratory chain, the mechanism that muscle cells use oxygen to create energy during physical activity. People carrying a mutation in this gene have VO2 max up to 50% lower, not because their lungs are weaker, not because their heart is smaller, but because at the molecular level inside each muscle cell, the ability to process oxygen is limited from before they were born. Cold activates the expression of PPRG-C1A, does not delete the mutation if present, but increases the level of gene expression enough to compensate for a significant part of that deficiency. PSME1 is mentioned less often in discussions about cold therapy, but its mechanism relates to one of the most important degradation processes in the body. Cells continuously produce proteins, and during that production, a certain proportion of proteins are misfolded, damaged by oxidative stress, or become useless after completing their task. Damaged proteins that are not cleared will accumulate inside the cell, causing dysfunction, and accumulating long enough will be linked to neurodegenerative diseases such as Alzheimer's and Parkinson's, where damaged proteins accumulate into plaques inside neurons. PSME1 encodes a component of the proteasome, the molecular machine inside the cell whose function is to identify and grind down damaged proteins for recycling. Cold increases the expression of PSME1, the proteasome operates more efficiently, damaged proteins are cleared faster, cells maintain better intracellular order. RBM3 and CF address a problem at an even more fundamental level. Before proteins are synthesized, DNA must be transcribed into RNA, and that RNA must remain stable long enough for the ribosome to read and translate it into protein. RNA is not a stable molecule. It is easily degraded, easily deformed by temperature and chemical stress. RBM3 and CF are RNA-binding proteins. They bind to RNA and protect the structure of RNA from damage, especially under changing temperature conditions. When RNA is unstable, the proteins synthesized from it are misfolded, and misfolded proteins are exactly what PSME1 has to clear. RBM3 and CRP operate at the layer before that, preventing the problem from arising at the root instead of cleaning up the consequences. Cold activates both of these genes, stabilizes RNA, synthesizes proteins more accurately, cells survive better under stress conditions. In addition, RBM3 and CRP also have direct anti-inflammatory and antioxidative stress effects, independent of their role in protecting RNA. ACTN3 is the final gene in this chain and is the gene that explains a phenomenon that cold therapy practitioners consistently report but is often labeled as psychological increased stress tolerance. ACTN3 encodes alpha-actinin-3, a structural protein located in fast-twitch muscle fibers, the type of muscle fiber responsible for explosive power in response to high loads. This protein is not only related to the mechanical strength of muscles, but also related to the ability to withstand physical stress and the speed of recovery after injury at the muscle fiber level. Cold activates the expression of ACTN3. The body becomes more resilient in the literal sense at the molecular level. When cold therapy practitioners say they feel they tolerate stress better, are less overwhelmed by pressure, recover faster from difficulties, that is not because they have learned a new psychological skill. It is because ACTN3 and the entire gene network just described are operating at a level that willpower cannot reach. Gene expression has been measured directly by RNA sequencing in clinical studies, and the results are consistent. The downstream impact of these gene expression changes on specific pathologies in humans needs longer-term studies to fully confirm, but the existence of those expression changes is no longer an open question. Noradrenaline released into the blood by cold shock does not stop at the muscles, does not stop at brown fat, does not stop at the immune system. It crosses the blood-brain barrier and reaches the prefrontal cortex, the brain region responsible for directed attention, processing complex information, decision-making, and emotional regulation. Noradrenaline acts on alpha and beta adrenergic receptors in the prefrontal cortex and increases neural signaling between neurons in this area, meaning information processing speed increases, the ability to filter noise and focus on important signals increases, and the mental fog, that feeling that the brain is not operating at full capacity that many people describe in the morning or after a long day, decreases. This is the mechanism behind the alert and clear-headed feeling, that most cold therapy users report right after getting out of the tub. It is not because the cold water wakes them up in a sensory sense, but because noradrenaline has just raised the operating capacity of the prefrontal cortex to a level higher than normal. But noradrenaline is only the first part of the neurological story. The second part is dopamine, and this is where cold therapy differs completely from everything else humans use to feel better. Cold immersion activates dopamine synthesis through the noradrenaline pathway, and dopamine levels increase not in the tub, but after getting out, lasting from 2 to 3 hours at levels higher than baseline. Not a spike, then crash. Not a surge, then drop below starting point like caffeine does after adenosine builds back up. Not artificial stimulation, then exhaustion like nicotine or any substance that acts directly on dopamine receptors. This is real dopamine synthesis. The body creates that molecule from scratch and releases it in a natural rhythm, not by forcing the system to release stored reserves and then leaving a gap afterward. To understand why this relates to depression, you need to know that dopamine and noradrenaline deficiency is one of the central molecular mechanisms of clinical depression. It is not a theory, not a behavioral observation, but the mechanistic basis of one of the most common classes of antidepressants today. SNRI, serotonin noradrenaline reuptake inhibitors, which work by keeping noradrenaline and serotonin in the synapse longer, instead of letting them be reabsorbed immediately. Cold therapy increases the synthesis of both dopamine and noradrenaline from the source, not by blocking reuptake like drugs, but by stimulating additional production. The mechanisms are not the same, but the end result in the nervous system, more dopamine and noradrenaline available at the synapses, is in the same direction. Calling cold therapy a natural antidepressant is not a metaphor. It is a description of the molecular mechanism. The final layer in this neurological part is what people commonly call willpower, but is actually biology. Each time you get into the cold water tub, your brain receives a controlled, clear, time-limited stressor. The HPA axis activates, cortisol and noradrenaline rise, the sympathetic system reacts strongly, and then, when you get out of the tub, the entire system recovers and adjusts below baseline. The brain learns from that cycle, not in an abstract sense, but in a very specific biological sense. The HPA axis becomes more efficient at activating and recovering, the stress response threshold is recalibrated, and the response to the next stressor in life, whether work pressure, conflict, or any stressful situation, is handled by a system that has been trained through cycles of controlled activation and recovery. This mechanism is identical to what high-intensity exercise and intermittent fasting do to the HPA axis. The same hormesis principle, controlled stress making the system stronger rather than weaker. In addition, ATN3 expression increases at the molecular level. Structural protein in fast-twitch muscle fibers is strengthened, and the ability to withstand physical stress increases literally within each muscle fiber. A person sitting in 10 degrees Celsius water for 2 minutes is not practicing willpower in the psychological sense. They are training their HPA axis through a controlled stressor, increasing the expression of genes related to endurance capacity and activating dopamine synthesis that will last 2 to 3 hours afterward. The result they call feeling stronger, more resilient, less overwhelmed by pressure, is not because they have learned a new attitude. It is biology changing in that direction under the effect of a repeated physical signal. Dopamine and noradrenaline have been measured directly and consistently in controlled studies. The linking mechanism between neurological hormesis and what people call mental resilience or willpower in real life is inference from hormone data and observed behavior, with no direct measurement at the neural connection level yet. But the direction of the evidence is consistent, and the mechanism is highly plausible. While the brain is receiving sustained dopamine and the HPA axis is being trained through each cycle of activation and recovery, the vascular system is undergoing its own separate adaptation process. Total cholesterol is the measure that most people know, but it is an outdated measure in the literal sense of the word. Not because it is wrong, but because it does not provide enough information to assess real cardiovascular risk. What is more important is not how much cholesterol is circulating in the blood, but which direction that cholesterol is going. ApoB is the apolipoprotein attached to LDL and VLDL, the lipoproteins that transport cholesterol from the liver into the blood vessel walls, which is the accumulation direction. APOA is the apolipoprotein attached to HDL, the lipoprotein that transports cholesterol from the vessel walls back to the liver for processing, which is the cleanup direction. An increased APOA to APOB ratio after cold therapy means the balance between those two directions shifts toward cleanup, less cholesterol being pushed into the vessel walls and more being pulled out. And this is information that total cholesterol never gives you. But cholesterol only begins to accumulate in the vessel walls when the vessel walls have already been damaged. And this is where homocysteine appears. Homocysteine is an intermediate amino acid in methionine metabolism. And when its concentration in the blood rises, it causes direct inflammation on the endothelium of the blood vessel walls, creating small lesions that cholesterol then sticks to and gradually accumulates into atherosclerotic plaques. Cold reduces homocysteine, reduces inflammation on the vessel walls, reduces the number of sites where cholesterol can begin to accumulate. And the result is reduced risk of atherosclerosis, not by affecting cholesterol, but by affecting the prerequisite condition for atherosclerosis to begin. Completely separate from both of the above mechanisms is what happens to the blood vessels themselves with each cold exposure. When cold water touches the body, peripheral blood vessels constrict strongly to reduce heat loss from the skin, pushing blood towards central organs. When you get out of the tub and the body warms up, those blood vessels dilate again, constrict then dilate, repeated with each cold exposure. Blood vessel walls are made of smooth muscle and elastic connective tissue. And just like muscles become stronger through cycles of contraction and recovery, vessel walls are trained through cycles of constriction and dilation to become more elastic, more flexible in responding to blood pressure changes and maintaining more stable blood pressure in stressful situations. Stiff, inelastic blood vessels are one of the important cardiovascular risk factors in middle-aged and older people, and this is a direct mechanical intervention into exactly that property. Insulin and uric acid complete the cardiovascular picture in two separate but related directions. Insulin decreases after cold therapy, and insulin sensitivity increases. These are two sides of the same improvement seen from different angles. Decreased insulin means the body does not need to secrete as much insulin to handle the same amount of glucose, reducing fat accumulation and reducing chronic inflammation associated with chronically high insulin. Increased insulin sensitivity means cells respond better to insulin signals. Less insulin is needed to achieve the same effect, and the self-amplifying loop of insulin resistance is interrupted before it starts. Decreased uric acid reduces the risk of gout, reduces chronic joint inflammation, and reduces oxidative stress in blood vessel walls. Because high uric acid is an independent source of free radicals that contributes to endothelial damage, exactly the type of damage that homocysteine also causes, but through a different mechanism. APO ratio and homocysteine have been measured in controlled clinical trials, with results consistent with the direction of improvement, but these trials are small scale and need confirmation at larger scales before the magnitude of the effect can be stated with certainty, going back to the 10 seconds at the beginning of this video. Wrist under the cold faucet, heart rate jumps suddenly, breathing contracts, goosebumps rise, shoulders shrug up. At that moment, you did not know the names of what was happening. Now you do. TRPM8 on the skin activates within milliseconds. Noradrenaline increases 300%. Brown fat begins burning glucose. PGC1 alpha initiates the creation of new mitochondria. TSH and T3 increase. The HPA axis activates, then pulls cortisol below baseline. Killer T cells increase, zinc increases, glutathione increases, homocysteine decreases, APO ratio shifts toward blood vessel cleanup. PSME1 speeds up the grinding of damaged proteins. RBM3 and CRP stabilize RNA. ACTN3 strengthens muscle fibers at the molecular level. Dopamine is synthesized and lasts 2 to 3 hours afterward. All of that begins in the first 10 seconds. 6 layers of mechanisms: sympathetic, nervous, metabolic, immune, gene, brain, and cardiovascular are activated by a single physical signal. No pill activates all 6 layers at once. No device does it through a mechanism this simple. The wellness industry sells you a $5,000 cold plunge tub with temperature control screens and a brand name designed to sound scientific. Your body does not need any of that to activate the mechanisms just described. It only needs the signal. The signal is cold. In practice, the starting point is 50 Fahrenheit or 10 degrees 1 minute, 3 to 4 times per week, totaling 11 minutes. Not every day, not 1 hour. A cold shower at the end of your bath is enough to start. No separate tub needed. No equipment needed. No change to your schedule needed. Gradually increase time and decrease temperature week by week, so the body adapts instead of being shocked too much from the start. The first 20 to 30 seconds are the hardest. Controlling your breathing during that period is the only thing you need to do consciously. The rest your body handles itself. No one is saying this is a cure-all. 11 minutes per week is a modest dose, and the effects are proportionate to that dose. Not a miracle, not a replacement for medicine, not an all-powerful solution. But 6 layers of real mechanisms, measured by RNA sequencing, by blood tests, by direct hormone measurements, all activated by the cheapest and simplest thing you can do for your body in the morning. Turn the faucet to cold. Hold the first 20 seconds. Let your body do the rest. If the mechanisms in this video are useful to you, subscribe to the channel so you don't miss the next videos. Each video is one layer of biology that your body is operating that you didn't know the name of. Thank you for watching until here. See you in the next video.